Hello, my name is Professor Hudson, and you're about to view a narrated PowerPoint lecture for Chapter 13, How is Gene Expression Regulated? In the previous module, in Module 6, Part 1, we talked about how the information in DNA is used to construct proteins. Now we will talk about how cells can control which proteins are made at a given time. So let's talk about how is gene expression regulated. Briefly, let's just review the process of gene expression. Gene expression leads to the formation of a protein product most of the time. The two steps required for gene expression are transcription, which occurs in the nucleus, and translation, which occurs in the cytoplasm at the ribosomes. One DNA strand can serve as the template for messenger RNA. Once the messenger RNA is made, it is processed before leaving the nucleus, and messenger RNA moves into the cytoplasm and becomes associated with ribosomes. tRNAs with anticodons carry amino acids to the ribosome and match with the codons in messenger RNA. Anticodon-codon complementary base pairing occurs. When this occurs, the amino acid will be transferred from the tRNA over to the newly synthesized protein, and the ribosome will continue moving along the messenger RNA until a stop codon is reached, and then the ribosome will fall off and the protein will be released. Oftentimes, proteins will be modified in the ER and Golgi before they become functional. When we talk about gene expression, we talk about how the information in your DNA can be transcribed through the process of transcription into messenger RNA, and how this information in the messenger RNA serves as the template for translation to occur to produce a protein. With this process, there are several different ways in which cells can control which genes are going to be transcribed and thus translated into protein. The human genome contains 20,000 to 25,000 genes. A given cell expresses or transcribes only a small number of genes at any given time. Regulation of gene expression may occur at the level of transcription, translation, or protein activity. Some genes are expressed in all cells, such as genes coding for tRNAs, since all cells require proteins. Other genes are expressed exclusively in certain types of cells at certain times in an organism's life, or under specific environmental conditions. For example, if you're lactose intolerant, at some point your body has stopped making the protein necessary to break down lactose in milk. This is an example of where a gene that may have been on, say when you were a baby or a young child, is now off as an adult. Let's talk about how gene regulation occurs first in prokaryotic cells such as bacteria, then we will talk about how it occurs in eukaryotic cells. This figure shows the Lac Operon. In prokaryotic cells, gene expression is primarily regulated at the level of transcription. Prokaryotic DNA is organized into units called operons, which contain functionally related genes. Each operon consists of 4 parts: a regulatory gene, which controls the timing and rate of transcription of the other genes, and note that in this figure; a promoter, which is the site for RNA polymerase to recognize and start the transcription; an operator that governs access of the RNA polymerase to the promoter. So the operator actually works almost like an on-off switch. And the structural genes, which actually encode for the enzymes or protein that the cell is trying to make. Let's talk about how the Lac Operon actually functions using a real-life example. The intestinal bacterium E. coli lives on what its host eats. Specific enzymes are needed to metabolize the type of food that comes along through the digestive tract. For example, in newborn mammals, E. coli are bathed in milk, which contains the milk sugar lactose. In order for the bacteria to break down the lactose, it must synthesize certain genes necessary for lactose metabolism. The lactose operon contains 3 structural genes which aid in the production of enzymes for lactose metabolism. The way this operon works is that in the absence of lactose, the lactose operon is shut off by the repressor protein. The regulatory gene of lactose operon directs the synthesis of this protein, which represses the operon by binding to the operative site. Under these circumstances, RNA polymerase, although it can find the promoter, it cannot transcribe the genes for metabolizing lactose because the repressor is blocking access to these genes. When lactose is present, lactose will bind the repressor protein and change its shape so that it can no longer interact with the genes for producing enzymes for lactose metabolism. When bound to lactose, the repressor is altered such that it does not bind the operator. Without the repressor and the operator, RNA polymerase is able to reach the promoter and begins transcription of the genes necessary for metabolizing lactose. When all of the lactose is gone, then no lactose will be binding the repressor. The repressor will assume its normal shape and then begin blocking the transcription again. So basically what happens in the Lac Operon, if lactose is absent, there's no need to make genes for breaking down lactose. So the repressor will be bound to the DNA and will block the RNA polymerase from making messenger RNA. If lactose is present, then we do want to make enzymes for breaking down lactose. And so that lactose will actually bind to the repressor, inactivate it so that it cannot bind the operator. Then messenger RNA will be able to make the necessary enzymes. So when lactose is absent, the operon is shut off. When lactose is present, the operon is on. This is typically how prokaryotic cells regulate whether or not certain proteins are made. It's typically at the level of messenger RNA transcription, and most of their genes are clustered in what we call operons. There are various operons for the different proteins that they need to synthesize. This figure is just illustrating regulation of the lactose operon in a different fashion. This figure, I believe, is the one from your book. The previous figure that you looked at was from another textbook. Now let's talk about eukaryotic cell regulation. In eukaryotes, gene expression is regulated at many levels, not just transcription. Although eukaryotic gene regulation bears some similarity to regulation in prokaryotes, the complexity of eukaryotic cells leads to differences as well as some similarities. Remember, in eukaryotic cells, the DNA is in a membrane-bound nucleus. There are a variety of cell types in multicellular eukaryotes. The genome of eukaryotes is organized differently. RNA transcripts undergo complex processing not seen in prokaryotic cells. Expression of genetic information by a eukaryotic cell is a multi-step process, beginning with transcription of DNA and ending with a protein that performs a particular function. Let's look at this figure. This figure illustrates some of the different levels for which you can have regulation of gene expression. Using this figure as a guide, regulation can occur at many steps along the process of making a protein. First, Cells can control the frequency at which an individual gene is transcribed. Regulatory proteins binding to a gene's promoter alter its rate of transcription. In eukaryotic cells, transcriptional regulation can occur at least three levels: the level of the individual gene, regions of the chromosome, or entire chromosomes. So even within this first level of regulation, when we talk about the frequency of transcription, there are three sublevels or additional levels within this level of regulation. As mentioned, at the individual gene, regions of the chromosome, or the entire chromosome. In particular, at the level of the individual gene, sometimes chromatin packing can play a role in determining whether or not a protein will be made. Then, once we move beyond this first level at which we control the frequency of transcription, we can also control the amount of protein that is made by, or the types of proteins that are made by taking the same gene and using it to produce different messenger RNAs and thus different protein products. Differential splicing of exons can yield different proteins according to a cell's needs. Both controlling the frequency of transcription and processing of the messenger RNA transcript are levels of regulation that occur within the nucleus. Controlling the frequency of transcription is transcriptional regulation, and controlling which messenger RNAs are made from a single gene also involves transcriptional regulation. Now, moving outside of the nucleus, cells may control the stability and translation of messenger RNAs. Some messenger RNAs are long-lasting and translated into protein many times. Others are translated only a few times before being degraded. Small regulatory RNA molecules may block translation of some messenger RNAs or may even target some messenger RNAs for destruction. This is sometimes referred to as post-transcriptional regulation. Basically, how long the RNA survives in the cytoplasm before it breaks down can control the rate at which a protein is made. Another level that occurs in the cytoplasm involves the fact that proteins may require modification before they can carry out their functions. Some modifications of the inactive protein involve excising a segment of it, exposing an active site, and activating it. Another common method of changing an enzyme's activity is phosphorylating or dephosphorylating it. Controlling these modifications provides a way to regulate the enzyme. This level is sometimes referred to as post-translational regulation. And then the last level, also seen in the cytoplasm, is that cells can control the rate at which proteins are degraded. By preventing or promoting a protein's degradation, a cell can rapidly adjust the amount of particular protein it contains. So basically, along the continuum from DNA to RNA to protein, we have 5 main categories for which transcription and translation can thus be regulated. 2 levels occur within the nucleus, and then there's 3 levels that can occur within the cytoplasm. In addition to the above, some other mechanisms of regulating gene expression exist. Epigenetic controls can alter gene transcription and translation. Epigenetics is the study of how cells and organisms change gene expression without actually changing the nucleotide sequence of DNA. This can involve modification of DNA, modification of chromosomal proteins, and changing transcription and translation through the actions of several types of RNAs, collectively called non-coding or regulatory RNAs. This will conclude our narrated PowerPoint lectures for Chapter 12 and 13.